Welcome to our senior secondary biology series. In this video, we're going to take a look at enzymes. This video or this topic is going to be presented in two videos. This is part 1 and there will also be part 2. So as usual, start with the learning outcomes. So at the end of this video, we should be able to define an enzyme. We should also be able to state the nature of an enzyme. We should explain the lock and key mechanism. So we're going to learn about a mechanism called lock and key. It explains how enzyme works. And finally, we should be able to explain factors that affect enzyme activities or the rate of an enzyme catalyzed reaction. Enzymes. So why are we studying enzymes and what are enzymes? So in studying life, in an effort to understand life, we have looked at the properties of life, and we looked at chemical reactions that occur inside living organisms, which are termed as metabolism. Those chemical reactions are possible because of enzymes. Of course, they can occur even without enzymes, but enzymes facilitate the processes that are involved with those chemical reactions. How does that happen? Let's start. So what is an enzyme? So an enzyme is defined as a biological catalyst. So biological means it's something that has to do with life or living. And what is a catalyst? A catalyst is defined as a substance that speeds up the rate of a chemical reaction without itself being changed in the process. So you can picture a catalyst as something that helps something to happen faster without it being really involved in the process. You can, okay, for example, what we're doing now, I'm your teacher, so you can picture me as a catalyst in the process of you acquiring knowledge. You can acquire knowledge, you can understand enzymes on your own, but I help the process to be fast. But at the end of the day, I'm not the one who's learning, it's you who's learning. So that's what catalysts are. They just speed up a process, but they don't actually participate in the process. So that's what an enzyme is. It's just a biological catalyst. So a substance on which the enzyme acts on is called a substrate. So the way enzymes work, it attaches itself to something called the substrate. Then it will modify the substrate so that the substrate, when participating in a certain reaction, the reaction will be faster. So that's what enzymes do. We're going to understand how that happens just in a few seconds. Enzymes have got different shapes depending on the substrate they work on. An example can be something like this. So an enzyme has got two most important parts. The first one is called the allosteric site, and the other one is called the active site. So what's the difference between the active site and the allosteric site? By the way, this is just an example of an enzyme. It doesn't mean that all enzymes look like this. I'm not even sure if there's an enzyme that looks like this. But regardless of the shape, it has got two major parts. The first is the active site, and the other one is the allosteric site. So the active site is where the substrate fits. So, like I said, the way an enzyme and the substrate work, they fit into each other or they lock into each other, as I'm going to demonstrate soon. So an active site, this is where the substrate fits. Then other parts of an enzyme, other than the active site, are called allosteric site. So for this enzyme, a substrate can be something like this. So this can be a substrate. So without an enzyme, this substrate is supposed to undergo or to participate in a chemical reaction. And of course, there should be results. A reaction is where two things are mixed and some magic happen. And at the end of the day, there will be products or new things are going to come out of it. So the way it is, when it participates in that chemical reaction, the reaction might be slow. So the enzyme helps to speed up the reaction. How does the enzyme help to speed up the reaction? What it does is it By the way, we're going to use, and I'm going to explain the lock and key mechanism. So in the same lock and key mechanism, for us to understand how an enzyme and the substrate work, this the enzyme acts like a lock and a substrate acts like a key. So a lock and a key, each lock has its own key. The shape of the key somehow is related to the shape of the lock, or the point where you insert the key in the lock. So this is the part where you insert the key, and this is the key. So this is how the lock and key mechanism work. So the substrate will come here like a key and will fit here. That's when the enzyme will act on the substrate. So what I'm trying to say is something like this. So you can see the substrate has perfectly fitted into the enzyme. So this enzyme is meant, just from the shape, this enzyme is meant to work with this substrate because of the shapes. So this is the lock and key mechanism. The enzyme acts as a lock and the substrate acts as a key. Then together they lock like this, forming what is called an enzyme substrate complex. So this is called an enzyme substrate complex. Once they do this, the enzyme will act on the substrate and there will be products. So the substrate will be something like this. So it will be broken if that will decrease or will increase the rate of reaction. So once the substrate is broken, the byproducts of the substrate are going to participate in the chemical reaction, and the reaction will now be faster. So that's what enzymes do. Once they lock with the substrate, they modify the substrate, making sure that once it participates in the chemical reaction, the reaction is going to be fast. That's how enzymes work, and this is the lock and key mechanism. During an exam, if they ask you to explain how enzymes work using the lock and key mechanism, you have to explain just like the way I've explained it. Of course, using few words. Like in the lock and key mechanism, the enzyme acts as a lock and the substrate acts as a key. And the substrate has got a shape that corresponds to the active site of the enzyme, so that when they act, the substrate will perfectly fit the active site of the enzyme. So this is how enzymes work. By the way, don't forget that at the end of the day, when the substrate is worked on, these are termed as products of this process here. So we're going to start with the nature of enzymes or characteristics of enzymes. How can we tell that this is an enzyme when you have been given a sample or something? Then they ask you, is this an enzyme? How can you tell? So the first is enzymes are proteins in nature. Proteins are things like meat. So when you think of enzymes, think of meat. Yeah, so what other characteristic? Enzymes are catalysts. If something is an enzyme, it should be able to catalyze a reaction. Third, they catalyze both forward and reverse reactions. So what does it mean to catalyze both forward and reverse reactions? So there are two types of reactions. One is forward reaction, the other one is reverse reaction. An example of a forward reaction is a reaction where you mix two things to get a third thing. For example, let's say cooking shima. Okay, mix water and milk. At the end of the day, you are going to get shima. That's a forward reaction. A reverse reaction is where the end product can be broken down into the components that makes it up. So in the exam that I had given of shima, so a reverse reaction is you get shima, then you do some magic. That shima, you make it into water and milk. So that's a reverse reaction where you get the things that made up whatever you have. That's a reverse reaction. So enzymes work on both forward and reverse reactions. Fourth, they are specific. A given enzyme can only fit to a specific substrate. So from the lock and key mechanism, you saw that the active site of an enzyme is somehow matched to the shape of the substrate. Therefore, a given enzyme can only work on a given substrate. Doesn't mean that one enzyme can work on different substrates. No, it only works on a specific substrate. There is a question that likes to come, which goes like, explain enzymes specificity using the lock and key mechanism. So you can just explain how the lock and key mechanism works and concentrate on how the active site is matched or has a shape which is matched to the shape of the substrate, making the enzyme or the lock specific for a given substrate or a given key. Also, by the way, forget about this note. Let's go to the next point. It's a mistake. So their activities are affected by substrate concentration, enzyme concentration, pH, temperature, inhibitors, and cofactors. Of course, most of the terms here are not familiar with them, but we're going to take a look at each of them in details. But one thing you can pick from here is the activities of enzymes are affected by various factors. In short, given an enzyme, it can work well when certain conditions are met, and it cannot work well when certain factors have been changed or when certain conditions have been changed. If a question comes like, explain the characteristics or the nature of enzymes, and you want to use this point, there's a memory aid on mastering these factors, and it's septic, where the S is for substrate concentration, E for enzyme concentration, P is for pH, T temperature, I for inhibitors, and C for cofactors. So let's take a look at the factors that affect enzymes activities in details. So the first is temperature. So let's say we've got an enzyme and a substrate like this, or maybe we have 3 substrates. So at a given temperature, this enzyme will be working in the substrate. It will be working on the substrates. So as you have seen, it worked on this one, then it moved and it went to work on this one and this one and this one, just like that. Of course, after it works on it, there should be products that should be formed. So when we increase the temperature, in nature, when you increase the temperature of the environment, the kinetic energy of the molecules or particles also increases. Kinetic energy is also called moving energy. So you increase the temperature, the kinetic energy increases, which means the particles will be moving faster. If the particles are moving faster, or the molecules are moving faster, or in this case, the enzymes and the substrates are moving faster, it means there will be more interactions between the enzyme and the substrate, because there are more movements, which means they are meeting more often, they are fitting more often, and that will result in the reaction or the activity being faster. So that's how temperature affects the rate of enzymes activity. So increase the temperature, the kinetic energy increases, the interactions between the molecules or the enzyme and the substrate increases, and therefore the enzyme's activity or the enzyme's rate of reaction increases. So with that being said, if you keep on increasing the temperature, there will be a temperature at which the enzyme activity will be at its highest. In short, the enzyme is going to be more active. Such a temperature is called optimum temperature. Most of the enzymes that work inside the human body have got an optimum temperature of 37 degrees Celsius. Why so? Because that is the normal temperature for a human body. So around 37 degrees Celsius, they work well. When you reduce the temperature, they won't work well. And when you increase the temperature, something is going to happen. So what happens if you increase the temperature beyond the optimum temperature or above the optimum temperature? So when you are cooking meat is protein, just like an enzyme is a protein. When you are cooking it, when you increase the temperature, as the temperature increases or the heat increases, the texture of the meat changes. So even with enzymes, that's what happened. If you keep on increasing the temperature, the shape of an enzyme is going to be distorted. It's going to change. An enzyme whose shape has changed is said to have denatured, and a denatured enzyme might look like something like this. So if this was the original enzyme, after increasing the temperature, it might appear like this, for example. So if it is like this, you can see that the active site has been changed, and the active site is the most important. So if the active site has changed, can the enzyme still work on the substrate? So if this enzyme tries to fit into the substrate, nothing will happen. They won't lock, mean that there won't be any activity. Therefore, at temperatures above the optimum temperatures, there is no activity, or the enzyme's rate of reaction decreases. To help us understand how temperature affects enzyme activity, or what temperature is related to enzymes activity, we're going to use this diagram. So on this scale, we've got temperature. So as we go this, it means as we go this side, it means we're increasing the temperature, and as we go up, it means enzymes activity is being increased. So we can see that as the temperature is being increased, the graph is also going up, meaning that the rate of reaction is also increasing. A point is reached where the enzyme activity is at its highest. Such a point, like I've said, is the optimum temperature. But if you increase the temperature beyond the optimum temperature, enzyme activity dies because the enzymes have been denatured and can no longer fit with the substrate. Another fact that affects enzymes activity is pH. pH stands for potential of hydrogen. But are we getting any sense from potential of hydrogen? So potential of hydrogen is just a measure of acidity or alkalinity of a solution. So solutions can be in three forms. They can be neutral, where they're not acidic, or alkaline. For example, water. Water is not acidic. When you're drinking water, it's not like you're drinking an acid or an alkaline. Then some solutions are acidic, which means the acids like lemon juice, the battery acid, I think you know it. Then some solutions are alkaline, like Boom detergent paste. It's alkaline in nature. So different solutions have got different pH value. The value of pH tells or gives you an insight into whether it's an acid, or it's an alkaline, or it's neutral. There's this scale which is called the pH scale, which is used to measure the acidity or alkalinity of a solution. So the way the pH is that's got numbers from 0 to 14. So numbers between 0 to 7 represents acidity. So let's say a situation where you have been given a solution, then you put your pH meter, or the instrument that is used to measure pH inside that solution, and it reads 4. You have to know that your solution is acidic, or it's an acid. So as you go down the numbers, the acidity level increases. In short, a solution that has a pH of 2 is more acidic as compared to a solution that has a pH of 4. The same is true for numbers between 7 to 14. Only in this case, it's alkalinity. So numbers between 7 to 14 represents alkalinity. Therefore, a solution that has a pH value of 8 is an alkaline. It's not an acid. It's the opposite of an acid, called an alkaline. A solution that has a pH of 11, it's more alkaline than a solution that has a pH of 8 or 9. Then there is the middle point, 7. So 7 represents a neutral solution. So if it reads 7, it means the solution is neutral. It's not acidic, and it's not alkaline. So that's how a pH scale or pH values are used. But why are we talking about pH scales and pH values? Because the pH of a solution affects enzymes activity. Unlike temperature, different enzymes have got different pH, optimum pH. So optimum pH is the pH value at which an enzyme works best, or pH at which enzymes activities is at its highest. Meaning that at that pH, the enzyme is working fine. If you change the pH, then the enzyme's activity will decrease. Different enzymes have got different pH values, or optimum pH, so to put it. For example, enzymes in the stomach have got a pH of, work around the pH of 1, 2, or 3. Why? Because inside the stomach there is acid which helps in the digestion of food. So the environment in which these enzymes work are acidic, and therefore their pH is also the acidic pH. There are some enzymes with an alkaline pH, like the enzymes in the duodenum. If you don't know what a duodenum is, you will very soon. Yes, so what we can get from here is different enzymes have got different optimum pH, and if you change the pH, their activity will decrease. We can use this graph to understand what I'm just trying to explain. So again, going this side, pH values, and this side, rate of enzyme catalyzed reaction. So for pH, each enzyme has got one pH at which it works best, the optimum pH. At any pH other than the optimum pH, enzyme's activity decreases, or it won't work at its best. Another factor that affects the rate of an enzyme catalyzed reaction is substrate concentration. We're going to use these to understand how substrate concentration affects the rate of enzyme activity. So let's picture a situation where we have 3 enzymes like this and 1 substrate. So this substrate will fit into this enzyme. These other enzymes are going to be dormant because there are no substrates to work on. So when this happens, the rate of reaction will be slow because the substrates are not many as compared to the enzymes. However, if we increase the substrate, let's say there are 2, it means these 2 substrates are going to fit into these 2 enzymes, and therefore the reaction will be faster because there are more substrates which are being worked on. So as you keep on increasing the number of substrates, the reaction is going to be increasing. But let's say we now have 4 substrates. So the first 3 substrates will fit into the first 3 enzymes, but this other one we won't have any more active site to fit in, so it will be dormant. So even if you keep on increasing the number of substrates, the rate of reaction won't increase. So when it comes to substrate concentration, you increase the substrate concentration, the rate of an enzyme catalyzed reaction or enzyme activity will increase. But there is a point where even if you keep on increasing the substrate concentration, nothing is going to change. We are going to use this graph to understand what I've just explained. So again, we have enzyme concentration and rate of enzyme catalyzed reaction. I think I made a mistake. It's supposed to be substrate concentration. So as you increase the substrate concentration, the rate also increases. But the point is reached where there are no more active site for substrate to fit in. So don't mind this. I think I used a wrong graph. This was supposed to be for the next point. But at this point, there are no more active site for substrates to fit in. So even if you increase the number of substrate, the rate of an enzyme catalyzed reaction won't change. So this is why this part is constant, meaning that as you increase the number of substrate, the rate does not increase. So in summary, you increase the number of substrates, the rate increases, but a point is reached where all active sites for the enzymes have been occupied by the substrates you have added. So adding more substrate won't change anything. So that's how substrate concentration affects the rate of an enzyme catalyzed reaction. The other factor that affects an enzyme catalyzed reaction is enzyme concentration. So just like the way we have studied substrate, the same is true for enzymes. So let's say we have this scenario. So if we have one enzyme and we have got 3 substrates, this enzyme will work on this. Once it's done, it will move to the next one. Once it's done, it will move to the next one. But what if we add another enzyme? There are 2 enzymes. This work on this one and this work on this one. So in short time, both of these will be ready to take part in the reaction. And if the number increases to 3, the reaction will even be faster because all of them are going to be dealt with at once. But increasing number of enzymes, let's say there are only 3 substrates, increasing the number of enzymes won't change anything because we just need 3 enzymes to work on these 3 substrates. So increasing the number of enzymes won't change anything, just like the way we discussed when it comes to increasing the number of substrates. So being a 4th enzyme won't be of any help. So like I said, I swapped the graphs. So this is supposed to be enzyme concentration now, and the other side is supposed to be rate of enzyme catalyzed reaction. So as you increase the enzyme concentration, the reaction will increase till a point is reached where all substrates have occupied the active site, and the additional enzyme is not going to do anything. Therefore, the reaction, as much as you increase the enzyme concentration, the action won't be increasing. So the graph will just be constant like this. Enough with graphs. Let's take a look at some interesting factors that affect enzymes concentration that don't have to do with graphs. The first, they are called cofactors. What are cofactors? A cofactor is defined as any substance whose presence makes an enzyme active. So a cofactor is any substance, if it's there, the enzyme is active or it's more active. So that's what cofactors are. So there are some vitamins which if added, it's vitamins. Yeah. So there are some vitamins which if added to an enzyme or to a reaction, the enzyme, that reaction will be active. So such things are called cofactors. There are 2 types of cofactors. The first are called coenzymes, and these are organic cofactors such as vitamins. So the word organic means things like us humans, we are organic. Lemon is organic. What else is organic? Meat is organic. So when you hear organic, just think of things that are living in, but of course those who study chemistry will understand what organic means. So coenzymes are organic cofactors such as vitamins. So vitamins are organic in nature and their presence makes some enzymes more active. The other ones are called activators. So activators are inorganic cofactors such as amino salts. So things that are inorganic are things like rock. A rock is not organic, it's inorganic. Even salt. So there are some enzymes which, if salt is present in the environment, they will be active or more active. So these are the cofactors are other factors that determine the rate or that affect enzymes activities. Let's look at the last factor. So the last factor are called inhibitors. So what is an inhibitor? So an inhibitor is defined as a substance that slows down or completely stops enzymes activities. Cofactors do the opposite, but inhibitors, they slow down an enzyme activity or completely shuts down an enzyme's activity. So there are 2 types of inhibitors. The first are called competitive inhibitors. So let's say we have an enzyme and a substrate. So this substrate is supposed to fit into this active site. So in the presence of a competitive inhibitor, this is an example of a competitive inhibitor for this enzyme. So a competitive inhibitor has got a shape just like the substrate, but it's not the substrate. Like if you see, it looks like the substrate here, but the other side is not. So you can, so because of that, it can fit into the enzyme. So instead of the substrate fitting, so the the competitive inhibitor fit into the active site. So competitive inhibitor compete with the substrate for the active site. The other types of inhibitors are called non-competitive inhibitors. So these ones, they don't compete with the substrate. What they do is they go on the allosteric site, then they using the allosteric site, they will change the shape of the enzyme. For example, they can make it smaller so that the active site changes so that the substrate does not fit. This marks the end of this part of enzymes. Watch out for part 2. Make sure you like, subscribe, and share if you're watching on YouTube or Facebook. Otherwise, happy solving.